So tonight, for this lecture, what I'm hoping to do is convince you that although you think you came here out of your own free will, actually free will is a bit of an illusion. And many of the things we do are maybe not quite as much in our possession as we thought. This is also partly an excuse. If you get up and leave halfway through, oh no, that was not free will either, and you've been motivated to do that by something else. And it clearly wasn't a poor lecture. So what I want to talk to you today about is the microbes that are in and on and around all sorts of organisms and are having much more subtle effects than some of the things we've discussed in the previous lectures in this series. So previously we've talked about microbes doing really subtle things like killing people. And tonight we're going to talk about some of the much more subtle things that we think are influencing our behaviours on a sort of day-to-day basis. And I've been talking about microbial manipulation for many years, but nowadays I don't have to do any real introduction anymore because, fortunately, big-time Hollywood has got in on the act recently, and some of you in this room, there are some people coming in, there are some seats at the front if you want to grab a seat. Big-time Hollywood has come into this in the last few months with this series, The Last of Us, which has anyone seen that so far? Some nodding yet in the room. So maybe the people who are listening online are actually watching it in parallel because it's more interesting. So this series, for those of you who are not familiar with it, is based on a computer game. And it's a story about humans being taken over by an evil fungus that turns us all into zombies. Happily, that itself is not based in fact. But the premise, the underlying premise certainly is, because the idea for this whole genre of games and TV series is a real-life microbe that manipulates us. And it's, in fact, a group of species of fungus called cordyceps, or ophiocordyceps more recently. And these are tropical fungi that live on insects. And the unique feature of these kind of pathogenic fungi is that they manipulate their host to their own advantage. So these fungal spores are picked up by a variety of insects, depending on the species. The one you have in front of you is an ant-specific fungus. The ant, in its day-to-day process, will pick up one of these spores and ingest the spore. The spore then hatches inside the ant, and it starts to grow inside the ant. And when it's exhausted everything it can get from the ant, it is about to sporulate and release its own spores. The fungus knows that it has a problem. Ants typically live on the ground or sometimes down in their burrows. If you sporulate inside a burrow, that's not a very effective way to spread. What you want to do is get a big airborne canopy. And so at that point, the fungus takes over control of the ant's mind, and that ant decides that it's time to go climbing, and it climbs and climbs and climbs the nearest available tree, and eventually at a certain point latches on, and you can see that here in this ant, latches on to a branch and then dies. And it dies now up high in the canopy. That allows the fungus to now emerge from the ant, and you can see that here, that's this extended process coming out of the ant, and release spores. And because that ant is now several meters up instead of on the ground, those spores spread over a much wider area than they would have previously. So this is a microbe that has manipulated its host for its own advantage in a really quite a cute and clever way, and quite a scary way. And this is, of course, why Hollywood loves it, because the idea that this might happen in people too is nice and gruesome and very exciting. I have to say, fortunately, we do not know anything like this in humans yet, and hopefully we never will, but you never know. So this is quite a famous and elaborate sort of manipulation, but actually a lot of microbial manipulation is much, much more simple than that. And we're going to start, I'm afraid, as I very often do here, with a tale of toilets, because much microbial manipulation involves things around toilets. So this, that you can see in this picture here, is Shigella. Shigella is a foodborne bacterial pathogen, a cause of very acute and quite severe food poisoning. And in fact, this is a sample of what we refer to as delightfully as bloody stool, i.e. bloody diarrhea from a patient. And you can see these little rod shapes in there, which are the bacteria shed by this patient. If you pick up Shigella, which you do from contaminated food, and eat it, it very rapidly sets up an infection in the intestine. And you soon know about it, because what happens is you get very potent, rapid, and uncontrollable diarrhea. And of course, the reason for this is because the bacteria is releasing a toxin, Shiga toxin, that is highly pathogenic to humans and triggers a shedding of your intestinal lining. So you start releasing a lot of fluid and blood. You get very rapid diarrhea. The reason the bacteria is doing this is because if you can't control your bodily functions, you start to defecate all over the place. And of course, these days, hopefully in the Western world at least, you can make it to a toilet. But over much of human history, and indeed still over much of the world, you won't make it to the toilet in time. And so the consequence of this is this. You end up with contaminated environments with human feces, which contain, of course, this bacteria. And it's relatively straightforward to see that once you have contaminated environments with bacterial feces, it doesn't take a huge leap of faith to imagine that you can rapidly then contaminate food and restart the cycle. This is a really unsubtle sort of behavioural manipulation, but it works very effectively, right? If you're a Shigella, you've got to get from one human to another. A really good way of doing that is getting a human to poop all over their next set of food and spread that disease very rapidly. And this is one of the reasons why this disease spreads, particularly in areas like refugee camps where you may have poor sanitation. A very effective, if slightly gruesome, way of behavioural manipulation that spreads the disease. It's to the bacteria's advantage. And I think that's a really important thing to remember, is that the reason that a lot of pathogens do things like create toxins is not just to make our lives miserable. They are in there because they have an advantage for the pathogen itself. So it's probably not that surprising that behavioural manipulation is also usually in the microbe's advantage. And there are some really spectacularly exciting examples of that we'll talk about later. One, though, that many of us have experienced over just the last couple of years, in fact, is much more subtle than that, and it's this kind of behavioural manipulation. If you pick up a respiratory virus, most recently, of course, COVID-19, but the same is true for lots and lots of respiratory viruses, common colds, influenza, rhinovirus, whatever, the symptoms you most typically get very early on are either coughing or sneezing. Okay? In both of those cases, these behaviours are incredibly advantageous to the pathogen. So this is an image, obviously, of someone sneezing. Typical human sneeze consists of about 40,000 droplets. Okay? Very interestingly, some of which go about 9 metres, which is approximately from me to the back row here. So if I sneeze in a minute, bad luck, all of you. So this is a big projection of material, 40,000 droplets. Each droplet in a heavily infected individual can have tens or sometimes even hundreds of viral particles. And in fact, this has been measured recently for SARS-CoV-2. And typically, although there's a huge variation between people, typically a single cough from a COVID-19 infected individual has about 100,000 live COVID-19 particles in it. So it's not a surprise that coughing and sneezing is a very, very effective way of spreading diseases around. Hence the long-standing poem about coughs and sneezes spread diseases. They really do. If you're a respiratory virus that doesn't trigger coughing or doesn't trigger sneezing, you're not going to do very well, and you will be very rapidly outcompeted by one that does. So this is a subtle behavioural manipulation in the virus's interests that happens every day, right? We all experience this. So those are fairly straightforward. It's not a huge leap of faith to realise that to spread yourself, causing people to have diarrhoea or sneezing or coughing is quite straightforward. But to me, the more interesting examples are the ones that get much more complicated and elaborate. And one of the most interesting areas for that is studying these pathogens that have multiple hosts. Okay, there are lots and lots of pathogens and parasites that require multiple hosts. One that we all think about if you travel in the tropics is malaria, for example. You know that malaria and many other pathogens are spread by mosquito bites because they have two hosts. They have a mosquito host and a warm-blooded mammalian host, and they've got to move between the two. And it's in this group of organisms where they're having to manipulate two or more sometimes separate hosts that we start to see the most interesting behavioural manipulations. And actually, it's one of the areas where we have learned the most about behavioural manipulation is by studying those kind of organisms. So let me introduce you to a few. Many of them have terrible names. I'm so sorry for that. There's no exam at the end of this lecture, you'll be pleased to know, so you don't have to remember them. But let me introduce you to this one first. This is Plagiorhynchus cylindriceus, trips off the tongue. It's here. It's an acanthocephalon, which is called a spiny, so there's a group called the spiny-headed worms, which is not very accurate because they're not actually worms, but nonetheless, they do have spiny heads, as you can see on this one here. And this is a fascinating group because they all pretty much are parasites, and they all have two separate and really quite unrelated hosts. In the case of this one, it lives most of its life in a woodlouse, okay? But it cannot complete its sexual cycle in a woodlouse. It needs to get from a woodlouse into a bird. In Europe, most of those birds are typically starlings. And it's got to complete this cycle. And so we have known now for many years that one of the most interesting things is if you go to an area where this parasite is abundant, starlings, and then you look at what starlings are eating, starlings are about 10 times more likely to eat an infected woodlouse than an uninfected woodlouse. And the reason for that is because when these guys get into a woodlouse, they convince the woodlouse that it's time to kind of turn into a party animal and start wandering around in the middle of the day, not hiding under leaves, going around in broad daylight, and essentially making itself very visible for the bird to eat. This is a parasite manipulation of the woodlouse that facilitates the parasite's transmission to the bird and then completes the cycle. And actually, this group of acanthocephalans are kind of masters at this. There are species that infect shrimp in water, and they convince the shrimp they should hang around at the surface of the water and cling to leaves, looking very appetizing to passing fish, because those need to go into the fish to complete their life cycle, and so on and so on. It's a group of many, many species, all of which have done something similar, but in different sets of hosts. And that, I think, actually is an interesting piece of biology, because you would, at face value, think that manipulating a shrimp and manipulating a woodlouse might be quite tricky to do. And yet what this tells us is that evolution has managed to devise both of those systems and many, many others in a relatively short period of time, which probably goes to explain that we're all basically at heart pretty much the same. And so a lot of these manipulations are very similar in different hosts, which is a theme we'll come back to in a minute. So the acanthocephalans are a neat example. There's a much neater one which I wanted to share with you. This is one of my personal favourites. Again, a really trippy, easy title. This is the parasite Leucochloridium paradoxum. I am no classic scholar, as those of you who've been to these lectures before know, but I think I can derive this from its Latin and Greek origins. So leuco is sort of white, chloro is kind of green, so it's a whitish-greenish thing, and paradoxum means we don't really understand it, so it's a whitish-greenish thing that we don't really understand, which is a very accurate description of this particular parasite. So this guy is a fluke, not as in good luck. A fluke is a type of organism, a trematode. And it lives in two hosts, snails of the genus Succinia, and then it has what's called a determinate host in a bird. We often talk in parasites that have two different sorts of hosts about intermediate hosts and determinate hosts. And what that essentially means, intermediate hosts are the ones in which they do lots and lots of replication. Determinate hosts are the ones in which they complete their sexual cycle. So you need both of these hosts to complete your life cycle if you're one of these parasites. So this guy lives inside Succinia snails. This is a normal snail. This is an infected snail. I think if you don't have to be a well-trained biologist to spot the difference between these two, it's fairly obvious that this guy at the bottom has got some pretty technicolor eye stalks going on here. What has happened is the fluke has been ingested as an egg by the snail. It's hatched, and it has started to migrate. It's migrated its way to the eye stalks of the snail, which are the things that do this in the end. And it has very delicately eaten away the optic nerve. So this snail is now blind, it can't see. And that is very advantageous for the parasite, because now the snail does not know if it's day or night, or if it's on the top of the leaf or the bottom. And so these infected snails start to wander around in the middle of the day on top of a leaf. For the parasite that wants to get itself into a bird, this is a very clever trick, not so great for the snail, but it's really good for the parasite, because now the snail is easy prey for the bird. But what I love about this parasite is it doesn't stop just there. It kind of goes one step further. And when it's sitting in that eye stalk of the snail, trying to attract the bird, it does so with a bit of disco dancing that you can see here, pulsating up and down, sort of looking for all the world like a little tiny caterpillar stuck on the head of a snail. And presumably, if you are passing, blackbird, this is just like a super attractive snack. You eat the snail. Unfortunately for the snail, end of story, but for the parasite, start of its new story as it moves into a new host. A very nice, neat bit of behavioural manipulation. So that's quite cute, and you'll notice a common theme in all of these stories about things getting eaten. But this one, I think, is one about which is the story gets even more elaborate. And here, for this parasite, there is a nice twist in the tale. So this is Dicrocelium dendriticum, another really easy name. I wish someone would go around and kind of just give these things common names. It would be much easier. It's another fluke, so it's relatively closely related to the ones you've just seen. This one, though, has a bigger problem because it has chosen, for reasons best known to itself in evolution, to have not 2 but 3 separate hosts. It needs to complete its life cycle to start off in a snail, to move from that snail to an ant, and finally from the ant to a sheep or another grazing animal, and then eventually come out in the feces of this animal and restart the cycle. So it has the same problem that those other parasites and pathogens have of trying to get itself from a host to a host, but amplified because it has now 3 hosts to get through. And so it has evolved some really clever trickery to do this. So if we start in the snail is crawling around and doing sort of snaily things. It will pick up one of these as a cyst or an egg in the grass. It will hatch, it will start to replicate inside the body of the snail, and at a certain point it will irritate the snail's lining sufficiently that the snail does the equivalent of, snail diarrhea or snail sneezing or something, and ejects the parasite in a big ball of mucus. Okay? Very successful. So now, lying on the grass is a ball of mucus with the parasite within it. This mucus is kind of sugary and sticky and is just the kind of thing that ants like. So a passing ant that sees this ball of sticky mucus will pick it up and ingest it. And now the parasite really goes to work because it enters the gut of the ant, it starts to replicate. And then once it's done that quite repeatedly, these can get up to several hundred parasites in a single ant. Then what happens is one of those individuals, only one, starts to migrate up the ant's body to the basal ganglion, the ant's brain. And once it gets there, it now tells the ant to start doing some pretty crazy stuff. What happens to that infected ant is it will go up the nearest blade of grass, right to the top of the blade of grass, clamp its jaws on, and then do a little bit of a handstand at the top of the blade of grass. Why is it doing this? It's doing this because if you need to get from him to him, the best trick you can possibly do is sit at the top of a juicy young blade of grass, because then along comes your innocent, unsuspecting sheep, nibble, 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 gets ant alongside its grass, completes the life cycle. That's pretty cute, but what is even cuter is if that ant is on top of a blade of grass and it gets a bit hot, the parasite is now in danger. If it gets so hot that the ant dies, the parasite dies with it. The parasite knows this, so if the air temperature rises to undesirable levels, it somehow releases its hold on the ant, and the ant then goes back down to the ground, starts wandering around, and presumably does the ant equivalent of kind of waking up after a late party in somebody else's house and wondering how you got there, and goes back to normal life. And then when the temperature cools down again, the parasite re-exerts its effect, and up goes the ant and does it again. So you have like a remote control ant. And I think this is the first instance we've seen of a parasite that really has a very elaborate level of control over the host. We don't know how it does that, but I think understanding this process could tell us quite a lot, actually, about this kind of subtle behavioural manipulations. So remote-controlled ants, sheep, and snails, all pretty cutesy. So we've heard a bit about things that get eaten, things that manipulate wood lice, all of these kind of things. And you're probably sitting there thinking, why did I come to this lecture? I'm a human, not happily an ant or a sheep. Surely none of this has anything to do with me, because as we know, humans have free will, and therefore parasite manipulation is totally irrelevant. Just as a completely irrelevant aside, actually, this film, Her Own Free Will, it was a 1905, something like that, early film, one of the early silent films, and is lost. No one knows the plot of this because the film has been lost. So we actually don't know whether she had free will or not, but let's assume she did, okay? So we all have free will. We don't get manipulated by things we think. But of course, actually, we know that's not really true. Because most of us grow up learning a little bit about infectious disease, and one of the ones we learn a bit about is this particularly unpleasant disease, rabies, okay? Happily not present in the United Kingdom, but still, of course, a problem across many parts of the world. And rabies, if you haven't come across it, is a lysovirus. It's a virus that replicates in the nervous system of animals. And it does that in order to manipulate its host quite dramatically. So typically this is a dog virus, and infected dogs, unfortunately like these ones here, display these very clear symptoms. Okay? The virus is transmitted by bites and scratches in saliva typically, and so what it wants to do is maximize its transmission from host to host. And so it causes, first of all, its infected host to salivate. It produces a lot of virally infected saliva. And secondly, it drives fear and aggression in its host so that that host is more likely to bite another host and spread the disease. And this is, of course, one of the reasons that if you travel to countries where rabies is still present, you're advised to stay well clear of any animals, particularly those showing abnormal behaviours. So here we have a virus that's triggering a whole bunch of things that are in its own benefit. It's getting saliva going, it's triggering anxiety, paranoia, confusion in its host animal, and unprovoked aggression. If you are unfortunate enough to be bitten by a rabid dog, you will go on to display exactly the same symptoms. Humans infected with this become confused, they start to hallucinate, they become aggressive sometimes, and they are essentially displaying the behaviours of the virus. And unfortunately, it's untreatable at that point, it's always fatal. But ultimately, this is a virus that is manipulating our behaviour in a really quite unsubtle sort of way. Despite the fact we have known about rabies for hundreds of years, and in fact it's been a source of great research activity for the last probably 150 years, we still don't really know how this simple virus manipulates the mammalian nervous system in such an elaborate way. Although recently we have seen some very interesting findings. So what you see here on this side of the screen is a protein structure of one of the proteins on the viral surface, the glycoprotein. And it turns out that a part of this protein from the virus binds to a nicotinic acid receptor, which is one of the neurotransmitter receptors in the human and indeed the dog nervous system. And it seems to suppress it. And that has a consequence of lowering serotonin levels in infected individuals. And you might know that serotonin is often referred to as the kind of feel-good neurotransmitter. People are told to do things like eat lots of bananas and chocolate because it gets your serotonin up and it makes you happy. And loosely speaking, that's true. So low levels of serotonin are correlated with depression and anxiety and some of the behaviours that we see in virally infected individuals. I suspect this is not the whole answer, but certainly there is some evidence here that the virus is actually quite targeted in terms of playing with individual components of the human nervous system to trigger these sort of weird and wonderful behaviours. So rabies, happily, is a disease that we don't encounter firsthand very much these days, and hopefully one day might even eradicate. But behavioural manipulation is not limited in humans to these really dramatic, let's all go and froth at the mouth and bite people kind of behaviours. There are much more subtle behaviours. And the one about which there has been the most interest in recent decades, and perhaps is the tool with which we might unlock some of these mysteries, is this guy here, Toxoplasma gondii. So this is a parasite. You can see it on the screen in front of you. That is sort of loosely related to things like malaria. And this parasite you might have come across, in fact, particularly if you're female and have had kids. So this is something you should be tested for if you're pregnant, or indeed preferably even before you get pregnant. It is not generally a problem for humans, although it's transmitted quite readily through things like undercooked food. But if you're immunocompromised, or if you're a very young infant, this can cause quite a serious disease. The big problem actually in humans is not to the person infected, but to their unborn infant if they're a pregnant female, because this is one of the few parasites that can transmit across the placenta. And if it does that, then the unborn infant can be very severely affected or indeed killed by this parasite. So we typically screen for Toxoplasma antibodies in pregnant women to understand whether they're at risk of this or not. Most cases, by far and away most cases of this though, are asymptomatic. You don't know if you're infected with this parasite because once it enters the body, it forms a latent cyst, so it goes dormant, typically in either muscle tissue or in nervous tissue. It's not dead, but it's silent. It's kind of hiding out, if you like. And that we have known for a very, very long time. And if you read medical textbooks, it will tell you this is an asymptomatic infection of no consequence. But recent data suggests that's not actually entirely true. So why is that? So here is it. It's quite an innocuous little thing, actually, zipping across the screen there. Why am I saying this is not entirely true? The reason I'm saying that is because we have learned quite a lot about this parasite and its manipulation of hosts in its natural host. Humans are not the natural host for this organism. Its natural environment is, like many of the things we've been talking about tonight, between two different sorts of host. It lives most of its life in a rodent, mouse or a rat typically, but it needs to complete its life cycle in a feline. So in this country, that's typically a domestic cat, but of course out in the rest of the world, it could be a lion or a tiger. Now, it's relatively easy to see, you've learned quite a bit, hopefully, so far, that for the parasite to get from here to here is quite straightforward in one sense, because as we know, cats really like mice, as you can see in this particular photo here. So I couldn't resist that. So cats quite like mice, and so on the one hand, being a parasite that infects mice and needs to go to a cat is quite clever, because at least one of your hosts is looking for the other one already. The problem is that the reverse is not true. Rodents don't generally like cats, and there's a very good reason for that. And so if you're a parasite, and they're quite good at evading cats, right? So if you're a parasite, it's not really great that your first host here doesn't get anywhere near the second host. And so the parasite has solved that problem because infected rodents lose their fear of cats. And in fact, heavily infected rodents start to seek out cats. They're attracted to cat urine, they hang around the cat litter tray, and they do all sorts of very unwise things if you're a small rodent. They have become resistant to fear, this particular sort of fear in particular. They are no longer scared of going around open spaces, and in fact, you can see that here. So here's a little video you can see at the top of a normal mouse. You can just about see the mouse, hopefully. He's in a big white box here. He doesn't like being in the big white box, quite sensibly. And he's hiding at the corners, okay? He's looking for a way out. He's doing a kind of mouse sort of thing. Those of you who've encountered mice in your homes or wherever, you know, they run along the corners, they hide in the skirting boards, they don't sit in the middle of the room waiting for you. But a toxoplasma-infected mouse doesn't behave like that at all. Look at this one down here. He's zipping all over the place. He's really happy, quite brave, happy to go out and about into the environment. Great for him, not so sensible if there's a cat around. And so these toxoplasma-infected rodents are significantly more likely to be eaten by passing cats, completing the cycle. That's bad news if you're a mouse, and kind of not so great news if you're a cat either. But what's it got to do with people? Well, it turns out, so in people, this parasite doesn't want to be in us at all. We're a dead-end host. But it gets into us quite a lot. And in fact, you can find this through all sorts of processes. So this is an MRI scan of someone admitted to hospital for another purpose, actually. And in that MRI scan, you can see here a small white blob. That is a toxoplasma cyst in the brain of this individual. Probably asymptomatic. I don't know why this person was admitted, car crash or a fall from a height or whatever. But the cyst was discovered there. And actually, some of you might have had this experience when you're I don't know, having an MRI for something else or sometimes a muscle biopsy, and they might come back and say, oh, you had a toxoplasma, and you think, gosh, you know, was that bad? And they're like, no, ignore it. However, so they get into the human bodies. We're a dead-end host, and for most of the time that we've known about this, we have assumed that was without any consequence. But it turns out not to be entirely true. So if you are latently, asymptomatically infected with this parasite, you're actually still significantly more likely to suffer from a variety of mental health issues. Schizophrenia, in particular, is significantly more common in people who are harbouring toxoplasma. And slightly more bizarrely, it turns out that the incidence of toxoplasma infection is about twofold higher in people who crash their cars, particularly if you've crashed your car in a way that was basically related to you driving like a lunatic. So if you run a red light, if you drive too fast, if you do those kind of things and crash your car, you're much more likely to have a toxoplasma. Why is that? We think that is because the parasite, although it cannot complete its life cycle in a human, is essentially treating us like a rat. Because as we know, humans are basically rats with less hair. So it's treating us like a rat, and it is trying to do all of those things that it does in a rat or mouse. It's trying to make you a bit brave, go out and about, not be afraid of stuff, and do unwise things like run red lights in your car. And so your behaviour is being manipulated by this parasite, because the parasite foolishly thinks that might help it end up in a maybe it will help it end up in a cat, but probably not. Perhaps, so these are kind of pathological things. Crashing your car is not great. Having schizophrenia is certainly not great. But one of the most interesting kind of facets of that has been much more recent data suggesting that this parasite might also be doing things that we think of as very, very human behaviours. So if you open your own business, you are significantly more likely to be toxoplasma infected than someone who doesn't open their own business. And the logic behind this is because opening your own business is risky, right? It might not work. And so the parasite maybe is helping you to take that risk when, if you're not parasite infected, maybe you don't. The unfortunate consequence is it might help you start your business. It's also more likely to help you go bankrupt, because you're also more likely to go bankrupt if you have Toxoplasma cysts in your body. All of this is quite cute and gets even cuter when you realise that in the UK, for example, about 30% of people are latently infected with Toxoplasma. So, for those of you in the audience here in the room, if you look left and right and then decide which of the three of you is carrying the parasite, you may wish to decide between you who's driving home tonight based on that. But the idea that fundamental human behaviours like risk aversiveness might actually be being influenced by hidden parasites, I think is a very, very neat and slightly disturbing idea. So Toxoplasma is one of the kind of things that we know the most about. We still don't know how it does this, what are the molecules it's using, what are the signals it's using to manipulate it, what are the consequences for the human nervous system. But we know quite a bit about it. But what I want to turn to now then is the stuff that we don't really know much about, but might, I think, if I had to guess, next 50 years, what's going to be the big story? And the big story, I think, is going to be what are we missing currently? What might be out there that is doing all sorts of interesting stuff that we have never thought about? And those of you who have come to any of these lectures previously will have heard me rabbit on at length about the microbiome. Okay? The fact that we are living, walking zoos full of interesting microbes. This is an image from a group in the US demonstrated where they looked at different body sites and quantified the different diversity of microbes living on them. You don't need the details. The point is we have microbes everywhere. The soles of your feet, up your nose, under your armpits, whatever. The balance of them is wildly different. The type of microbes living between your toes are different to the ones living between your fingers. Most of them, many of them, we know nothing at all about beyond the fact that they are there. And we don't know what they live on, what they produce, what they do. And the big black box, if you like, the one that we've talked about multiple times in this series previously, is the stuff that lives inside you. And for those of you who missed earlier lectures, your gut microbiome, the range of organisms inside your gut is extraordinary. Numbers vary wildly, but whichever way you cut it, we undoubtedly have more bacterial cells in our intestine than we do human cells in our entire body. So to a first approximation, you are definitely not human, you're probably microbial. And that's just the bacteria, right? There are fungi, there are viruses, there are parasites, there are all sorts of interesting things stably living in your microbiome, point one. Point two is, it changes crazily. You eat a sandwich on your way to this lecture, you've changed your microbiome. You go to the toilet at the end of the lecture, you've changed it again. When you're born, you acquire a microbiome from your mother. It then evolves over your infancy. All sorts of things happen to the microbiome. And we are already familiar, hopefully, with the concept in the kind of popular press that the gut microbiome might be really important for things like gut health, for, if you have ulcerative colitis or some of these conditions, gut microbiomes are really important. What might be about to break, though, and if I had to gamble, I would say this is the role that it may be having on our minds also. Why do I say that? Why might the things growing in your gut have an impact on your mind? Well, first of all, the first reason for saying that is really good experimental data from other models, and particularly from mouse models. So let me introduce here, this is a mouse, you can just about see the mouse here, and this is a typical behaviour of mouse. Mice, we've heard about mice a lot this evening, and I'm going to hear some more now. Mice are scared, right? Mice don't typically come out and about. They're not very sociable creatures, at least with humans. If you raise a mouse that's so-called germ-free in a sterile condition, so the mouse has no microbiome at all, is significantly more kind of nervous, it has different traits. It's described, I love this, it's described by animal technicians as being slightly aloof in its interactions with other mice when it has no microbes. If you then transplant in microbes from a normal wild-type mouse, it starts to become a little bit more confident. If you change that kind of ingredient list, the types of microbes you transplant, and in particular one called lactobacillus, if you have more lactobacillus, the mouse becomes even more confident and starts to be much more outgoing. Probably not wise if you're a mouse, actually, in some senses. But you can manipulate a kind of pretty obvious behavior around how anxious that mouse is by the contents of its microbiome. Most interestingly, I think, is recent data in another rodent, this is a rat, obviously, And in rats, there's been a lot of work done on mental health of rats, mental health of humans modeled in rats. You can induce a state that is like depression in a rat by stressing it out, effectively. If you stress out a rat enough to create a type of depression, not all rats will do this. So you have a bunch of rats. Some rats, in response to that stress, will develop behaviours that look a little bit like depression in humans. They become lethargic, they go off their food, they don't want to go out and about. Not all rats do that. If you then take the microbiome, so a sample of bacteria from the depressed rat, and transfer it into a rat that has not had any prior experience, that rat on its own does not become depressed, but it then will become very rapidly depressed with a relatively low stress burden, much lower than the first instance. And conversely, the other way around, if you transfer a healthy microbiome to a rat that's looking depressed, it will buck up. Okay? So here, there is a kind of a real-world example of something as subtle as kind of depressive mood being quite strongly influenced by the microbiome. The most interesting thing, though, about that, I think, is it on its own, it's not enough. So you can't just transfer the microbes and voila, you've triggered depression or you've cured depression. And I suspect the same will be absolutely true of humans. It's about the context that happens. So you need that transferred microbiome and also that secondary trigger of animal technician stressing you out or whatever to trigger the full effect. But nonetheless, the microbiome is predisposing you one way or another to respond. So how might that actually happen? What might actually be going on? Studying this in rats, mice, or let alone humans is really tricky because we have very, very complex microbiomes with thousands of species, billions of cells, really tricky. So people have started recently to turn to much simpler models, and one that just very recently has shown some really, really interesting results, I think, is one of my favourite laboratory models. This is the nematode worm Caenorhabditis elegans. It's going to swim across the screen in front of you. It's a microscopic nematode worm, about just under 1,000 cells in the body, so it's a very simple model, but a really powerful model for understanding lots of things. Nematode worms eat bacteria all the time, and so they have a gut microbiome, just like we do, with bacteria that's a mixture of the stuff they're eating and digesting and stuff that is permanently present there. What has recently turned out is that one of the organisms that they can come across is a bacteria called Providentia. Okay? And Providentia will occupy the intestine of the worm and live there, just like our normal gut microbiome. Whilst it's living there, it turns out that that bacteria is producing a molecule called tyramine. And tyramine is converted by the worm into another molecule called octopamine, which is a neurotransmitter. So this bacteria is essentially providing a source of neurotransmitter to the worm. And what turns out to be quite cute is that neurotransmitter crosses the border into the worm and starts to alter the behavior of its neurons. And the very, very clever thing about this is that by doing that, that neural signal encourages the worm to go and eat more Providentia. And so it's seeking itself out through this manipulation. And this is one of the first examples we have of a real molecular handle. It's a single bacteria producing a single neurotransmitter and triggering really quite an elaborate behavioral choice in the worm, what bacteria is going to eat next. This is a worm. We don't have octopamine signaling, so this is probably not happening in us, you'd be pleased to know. But what has turned out to be quite interesting is that although we don't have octopamine, we have an awful lot of bacteria in the gut that produce this molecule. This is gamma-aminobutyric acid, or GABA. It's a very, very widespread neurotransmitter in the human immune system. And broadly speaking, it's a kind of shutdown neurotransmitter. It silences the behavior of neurons. And it turns out that this molecule is made by an awful lot of gut microbiome inhabitants. It's also broken down by quite a lot of bacterial species. So the amount of GABA that's in your intestine at any one time can be wildly fluctuating, depending on whether you've got more makers or more breakers of this molecule. We know it can cross the intestinal membrane, and although we haven't demonstrated this, I think it's really quite plausible that the level of this neurotransmitter in your gut will be affecting the neural signals in the rest of your body, including perhaps influencing things like mood, appetite, and other behaviours. We're a long way off really knowing how that actually works, but I think it's a really nice example of a potential molecular mechanism that gets us from theoretical correlations between the microbiome and actual behavioural interventions. So all of this is theoretically possible. It might be influencing X, Y, and Z. Is there any real evidence beyond those mouse and rat studies that it really is doing something? And I think the short answer to that is yes, it's starting to come. There's a really interesting study quite recently done by colleagues at Oxford and other universities where they have taken a lot of people, these are not the people they took, but a lot of people, and they have analysed the microbiome of those individuals, so the diversity of bacteria and fungi in their guts. And those people have also filled in questionnaires that are designed to probe their characters for all sorts of interesting traits that psychologists measure us on, like, for example, how outgoing are you, how reserved are you, how likely are you to make new friends, how fearful are you of that kind of cocktail party scenario, so on and so forth. And it turns out that there is a really interesting link with the microbiome, which you can see on this graph here. So, for example, if you score higher on sociability, you're the kind of person who likes to go out and meet new people and have a cocktail party or whatever, you have significantly more of this kind of bacteria, Akkermansia, than someone who's much less sociable. Okay? Conversely, if you have what people call neurotic tendencies, as psychologists use this kind of catchphrase for, you're a bit fearful, you tend to think other people are out to get you or whatever. If you have more neurotic tendencies, you have significantly less Carinobacterium than someone who's not so neurotic. Now, the big caveat here, of course, is that we don't know cause and effect. Maybe being much more sociable means you go and eat different stuff with different people and you pick up more Akkermansia. That's possible. But I think together, there's quite a strong suggestion here that the relative balance of different bacteria in your intestines may be influencing things as subtle and as apparently human as our propensity to go and socialise. And as we get more and more data on this, which is happening very rapidly now, I think we'll see more insights in here. And you might be aware, in fact, some of you might have done this, you can now have your gut microbiome commercially sequenced. If you so wish, you can send off a poo sample and a company, for a small amount of money, will come back and tell you what you've got. My slight caution there is, of course, they might tell you what you got on 6 p.m. on Tuesday. By 9 a.m. on Thursday, it might be wildly different. So don't take too much stead by it. But that huge amount of data that is pouring in from people's gut microbiome, large-scale studies of behaviour and genetic studies of the host, I think will lead us within a relatively short time to start to make interesting predictions around things. And who knows, maybe even intervene. And I think, the prospect of intervening to make someone more sociable is quite terrifying. But the prospect of intervening to deal with things like depression, I think could be a real game changer for medicine. So these are some of the various behavioural interventions that microbes do. There are lots more to discover, I think, but I want to end just with a slightly upbeat message. It's slightly depressing, I think, at this stage to think, well, maybe nothing I do is really me. It's all about microbes instead. And just to reassure you, I don't think that's really true. So let me end with this rather nice. I'm quite a big fan of Turner paintings. This is a great Turner painting. This is Hero and Leander, and if anyone knows your Greek mythology, so Hero and Leander, they were lovers. They were separated by a sea, and every night Hero would wave her lantern down here, and Leander would swim across the sea to see her, and then he would swim back again. And then one night in this myth, the candle blew out. Leander got lost at the sea, and he was drowned. And when his body was discovered, Hero jumped in and killed herself too. So this is a great romantic tragedy. Why am I showing you this here? I'm showing you this to reassure you that I don't think great romantic myths are written as a result of bacterial intervention. I don't think that Turner's genius to create this was driven because he had a parasite in the brain. But it might be worth thinking about the fact maybe Turner's flamboyant genius was a little bit influenced by his microbiome. And maybe whether we like paintings like this or we like stories like that or not might not solely be down to your personal feelings, but also due to whatever you ate that morning and influenced your gut microbiome. Let me leave it there and take any questions. Thank you very much.
Professor May, thank you very much.
Thank you.
We've got time for a couple of questions, so let me start off with the room. My colleague's got a roving mic, so if you put your hands up for me if you've got a question.
I wanted to, so a lot of your specific manipulation examples involved behaviors that the microbes are doing that they had sort of particularly landed on that were suited to their life cycles. I was wondering in the human situation with the mass complex of organisms, are they all also there for a particularly adapted reason, or is there some element of randomness in terms of what neurotransmitter they happen to secrete that happens to fit closely into one of our receptors, and not to mention also, is there any like coordination among on a system level of the microbes in an organism?
Thanks. Yeah, those are really great questions. So first thing to say there, I think you're absolutely right that the almost the simple scenario of a parasite in its host is kind of the ultimate part of simplistic behavioural manipulation, right? If you need to get from A to B, anything that helps you get from A to B faster is good. And so those very straightforward manipulations like let's get my host eaten are much more common in pathogens. In commensals, so organisms that live with us, like the gut microbiome, it's much less clear whether there is an advantage in terms of that host manipulation. And I suspect the short answer might be not in many cases. Many of these so the GABA that I mentioned, this neurotransmitter that's produced by bacteria, there's no evidence they're doing that because it works on humans, but this is a widespread molecule that is of benefit to the bacteria itself. Of course, there is a slight follow-through, though, that you might produce this for a completely different reason if you're bacteria, but if it then triggers a behaviour that is going to help you spread, that evolution will select for that behaviour. And so if you want to really push the boat out, and that's the fun thing about Gresham lectures, right? I can speculate wildly now. Imagine a bacteria producing a neurotransmitter that makes someone more sociable. Therefore, you host more parties, you cook more food, you spread that bacteria more successfully to the people you're entertaining. The bacteria has done very well. And so actually there is an indirect selective pressure for that very subtle level of manipulation. I don't think there's any evidence that that's what's happening now, but it's at least a theoretical possibility that some of the things we do start to become interesting. And of course, you can go wild there and think about partner choices and sexual behaviour and all sorts of things that we do that spread microbes around. So who knows? You'll look in the mirror later and think, am I doing this because it's me or not?
I had heard, and maybe this is wrong, that there are some gut bacteria that are bad for people, that make it difficult to digest, that make you fat, whatever. I mean, wouldn't that be kind of counterproductive for the bacteria, sort of?
Yeah, that's a very good question. So there are two things. There are definitely gut bacteria that are bad for you. So you think about, so for example, Shigella that I mentioned, cholera, lots of things that are really, really bad for you. I think what you're getting at more is the more kind of the commensals. There are patterns of microbiomes that are associated with disease. And in fact, quite good evidence that things like irritable bowel syndrome, people have a different sort of microbiome. And indeed, in some cases, changing their microbiome will help deal with their symptoms. So the question is that good or bad for the bacteria? It's not necessarily bad, because of course some of the things that are bad for us as humans are quite good for the bacteria, like diarrhea, like poor dietary control. Those kind of things might be advantageous to the bacteria. And it's all about the relative balance. So if this is something that bacteria does that is beneficial to the bacteria and happens to be bad for you, it will probably still be selected for. The interesting question becomes whether there's a behavioural manipulation that is bad for us but good for the pathogen. And I think that is potentially possible. Of course, if it's so bad for us that we die and the bacteria die with us, it's going to get eliminated by evolution. But it's certainly true, as a rather long-winded answer, it's certainly true that there are microbiome patterns quite strongly associated with disease. None of those that I'm aware of yet are specifically linked to a particular behaviour, but it's theoretically possible they could be, definitely.
I've just got one from the online audience for you. Is there treatment for toxoplasma? You may have mentioned it, but...
Is there a treatment for toxoplasma? So there are two different questions there. So acute toxoplasmosis, so when the parasite's really growing and causing disease in people, there are treatments. It's a bit tricky, but there are treatments for that. The asymptomatic, so if you have a latent cyst, I mean, theoretically, you could probably do things to that patient to remove that asymptomatic cyst. Those things that you would do are actually not in themselves very good. So I don't think there's any reason at the moment that if for those of you out there thinking, I'm one of those 30%, don't panic. I would not recommend trying to get rid of your asymptomatic cyst because it's far, and I should have made that clear, it's very unlikely that just because you have a cyst, you will become schizophrenic, for example, but you are slightly more likely to be schizophrenic. So there's no kind of medical justification for treatment. They do exist, but at the minute, the risks would massively outweigh the benefits. When it comes to other pathogens, I mean, the idea of tweaking your microbiome for benefit starts to become more interesting, but it's in its infancy, that I think, at the moment.
Read of outbreaks of mass hysteria caused by fungi. Do you think that is a form of manipulation, or it's just an accidental symptom?
Right. So at the moment, mass behaviours caused by fungal infections are still the stuff of science fiction, happily. There is, though, an interesting question about mass behaviour. And we know that, right? For lots of human behaviours, you can think about things like kind of cult behaviour that we sometimes come across, where people do all sorts of bizarre things, sometimes even mass suicide, right? How much is that likely to be to do with their microbiome? I mean, I suspect the answer is very, very little. But I wouldn't rule out the fact that we might. I really don't think there's a parasite out there that's going to spread through this room and we're all suddenly going to go and do something bonkers later. But the idea, for instance, that you might have a particular sort of microbiome that predisposes you to think that the world is out to get you, or the world's about to end, or there's it's flat, or whatever, any of these other conspiracy theories, is not beyond the realms of possibility. I don't think there's a parasite that makes you think the world is flat, but there might be a parasite that makes you more skeptical of kind of scientific evidence, and therefore you're more likely to think about the world being flat, and you club together with other people like that, and you self-aggregate. It would be a very interesting study to do the sort of microbiome of people who belong to kind of crazy cults. I suspect we might struggle to get people to register for that, but if anyone out there is in a crazy cult and wants to volunteer, let me know afterwards.
There's a difference between in your microbes, about viruses and bacteria. Now, the viruses, like influenza, for instance, when it strikes the body and the person succumbs and dies, is that the end of the virus? And in the case of bacteria, of course, it will continue living. So what happens to it?
Yeah, so you raise a very good point there. The key is the transmission route of the organism. So influenza is a good example. Influenza requires a human cell to replicate. If you die, it'll be viable for a few hours, but eventually it will go. There are, of course, viruses that are very successful at spreading from dead bodies. Ebola is a very good case in point, where it triggers this bleeding, it's blood transmitted, and actually a lot of Ebola transmission is during funeral rites, where people are touching the dead body and picking up tissues. So it's not a given that the end of the host is the end of the organism. And for some bacteria, as you mentioned in particular, they can be very, very separated. So anthrax can live for 50 years without a host in the soil, no problem. So death is not necessarily a problem, and actually it could be an advantage. There's quite an interesting sort of bit of speculation around some organisms that might feed off the decaying body. So I work on a fungus called Cryptococcus that is good at eating decaying bodies. And so there's some idea that maybe actually killing hosts is not a bad thing, because it gives itself a nice little nutrient store for the next 6 months to kind of live off. So they're not specifically linked. But you're right that generally most pathogens don't want to kill their host, certainly not fast, because by killing your host fast, you reduce your ability to propagate. But there are good reasons why you might still need to kill your host to facilitate transmission, for example, from blood or other bodily fluids.
Do you think that we could genetically manipulate microbes that use neurotransmitter to manipulate as such in a good way, such as we lose fear just as Toxoplasma gondii on mouse?
Sorry, I missed the second bit. So do I think there's neurotransmitters?
Yes, the neurotransmitters that they pass to the mouse.
Oh, for Toxoplasma. So do I think it's making a neurotransmitter? That is certainly possible. And I mean, as in the bacterial example, a lot of people often think, neurotransmitter must be a really, really complicated molecule. Actually, a lot of neurotransmitters are quite simple molecules. So it is certainly reasonable, I think, that Toxoplasma and other parasites might be making those kind of human active neurotransmitters. As far as I'm aware, so Toxoplasma is very intensely studied, so I guess a little bit of me thinks that if it was that easy, we might have discovered it by now. But of course, the nervous system is incredibly complicated, so it's quite possible it's making something that is not itself a neurotransmitter, but is maybe processed into one or inhibits one that's already there or something. There are lots of subtle ways to manipulate it, definitely possible. And certainly for other microbes, I think there's now very good evidence that many, many bacteria make human active neurotransmitters, and that's probably a big part of this dynamic.
Thank you. These microbiomes that cause behavioral changes, are they due to some dark evolutionary development, or purely accidental?
Dark evolution is quite a scary concept, isn't it? Yeah, so I mean, essentially all evolution is accidental, right? Stuff happens, we mutate something, and then essentially evolution works by selecting the things that are advantageous and killing off the things that are disadvantageous. So I definitely don't think that all these manipulations have arisen as some kind of orchestrated plan or some kind of evil power that's trying to take us over. What has happened, so in the case of Toxoplasma, is perhaps the simplest. You can imagine that the ancestral Toxoplasma did not manipulate rodents. Something happened, some mutation happened, and it happened to produce perhaps a neurotransmitter, and therefore that parasite's mouse was more likely to get eaten. Now there's a massive advantage. That parasite is selected, it does much, much better. The other lineages go extinct, and that is now the one that it's worked on. And I suspect something very similar for the microbiome as a whole, where you've got these things that might be very subtle, but of course on a scale, even if you increase your likelihood of transmission by 0.001%, but you do it in 6 billion humans, you very rapidly have a major natural selective advantage. So yeah, I don't think it's a sort of dark orchestrated plan, but I do absolutely think that natural selection is selecting these things for their power to do that.
Fascinating talk, thank you very much. Just wondering whether fecal transplants really work if your biome changes all the time, and if you transplant a different fecal thing into your body, would it reject it or would it accept it?
Yeah, very, very good questions too. So fecal transplants, for those of you, just to lower the tone a bit more this evening, for those of you who haven't come across them, is the idea that you could take someone's healthy gut microbiome and infuse it into your own intestine, replace your microbiome, and you will get health benefits. So there is really, really good data that works for some selective situations. So there is some evidence for Crohn's disease, for example. There is very good evidence that if you have a Clostridium difficile infection that is not shifted by antibiotics, that replacing the microbiome can help in that instance. And there are various trials underway for things like ulcerative colitis and other gut health things. And I suspect the answer will be yes for those conditions. You're absolutely right that it is not trivial to make that microbiome take. So quite often you put in the microbiome from the donor, having got rid of all the bacteria in the host originally, and it still doesn't stick, if you like. We don't really understand the reasons for that. We suspect it's something about the interface between your immune system in the gut and those bacteria coming in. They're recognized as foreign and ejected, but we don't quite know that. Do I think you could start to manipulate people's behaviour with this? That starts to get very interesting. And you might be familiar with the fact there was a court case, for example, in the US at the moment about someone who received a fecal transplant for disease purposes, but then afterwards started to get obese. And it turned out that the person they'd had the fecal transplant from was obese. And so they are saying this, you've given me a dodgy drug here and I'm obese because my microbiome is doing it, not because I'm eating too much. And that starts to get to a very, very thorny issue of rights and responsibilities. But you can easily imagine a scenario where we might want to pre-screen fecal donors for their mental health, for example, because you probably don't want to have a microbiome that might be more associated with, paranoia or anxiety or something. There are lots and lots of tricky ethical grounds there. And at the minute, I think the science is way behind that. But I could imagine that 20 years from now, someone might be standing here saying, God, of course we do that. Wait and see, I guess.
Thank you so much for your questions, everyone. The last lecture in the series, A Microbial Future, will take place on the 10th of May. So we hope you can all join us then. Thank you very much, everyone, and thank you for coming.
Thank you very much.